Good afternoon. This is video 10. You could almost consider it video 9B because it basically builds off of the last one a lot. And at the end of this video, we're calling it video 10. You ought to be able to do 2010 number 1 and 2018 number 2. So we've seen this picture a few times back in the cell membrane unit, the cell communication unit where we're at now. We also saw it with endocrine recently. And so what was the biggest difference between these two pictures? There's a lot of differences, but the difference that we were trying to emphasize was the location of the receptor. This receptor is stuck in the cell membrane. So you might see that in a book called an I-N-T-E-R, intercellular receptor, because that means between cells. You can send and receive, receive information from between cells, and then, or a membrane-bound cell, a membrane-bound receptor. And then here you'll notice the receptor is inside the cell. So this is an I-N-T-R-A, an intracellular receptor. Intra means within. So like interpersonal skills for interviewing for a job is how you relate to other people. If I'm a good teacher, I should have decent interpersonal skills. Intrapersonal skills, I don't even know if that's a word, but that would be like my own mental dialogue within myself. Internet, between networks, intranet, within Keller ISD. There are things that I can't access here at home that I have to be at Timber Creek to access because it's on the intranet. Okay, so what do we know about that particular hormone, testosterone, versus this ligand, this hormone? Well, the basic idea is what has to be true about this, just like we saw in the previous videos, is testosterone is going to have to be small enough and non-polar enough to squeeze through that cell membrane. If I have something that's pretty large or pretty polar, then now that signal molecule, that ligand, that hormone maybe, could bind to a membrane-bound receptor like this. Generally, whenever these guys are able to pass through, they hit the receptor and they immediately go inside. So if this guy was able to pass through, so let's say there's my ligand, it hits the receptor, it'll go in and act as maybe a transcription factor to turn a gene on. So you might use it to turn a gene on or turn a gene off. However, and these are much more common, however, if it's a membrane-bound receptor, you usually go through 3 steps. As I've said before, I kind of call it 4 steps. So first you have to send out a message, then reception, then transduction, then a response. So that's what we're seeing here. We're seeing 2 different membrane-bound receptors. This one has a ligand that maybe looks like this. This one has a ligand that looks like that. So maybe that guy was really polar, so he couldn't get through the membrane like this one could. And essentially what will happen is whenever this ligand hits that receptor, the receptor changes shape slightly. That activates this protein, which activates this one, which activates this one, which activates this one, which might land on the promoter. That's the beginning of a gene. We'll be getting into molecular genetics in future videos, but you might remember like regulatory sequences, promoters, and then the end of a gene, a terminator. Same thing over here. Whenever this ligand hits this receptor, activate, 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 activate. Now, one thing that I'm not showing in this picture is amplification. So when this protein turns on this protein, turns on this protein, turns on this protein, or activates it, it might activate by adding a phosphate. So I might have these 3 proteins in a row, 1, 2, 3, square, triangle, star. And essentially what will happen is if this guy gets activated, he will go and phosphorylate. He will add a phosphate. He'll break an ATP, add a phosphate to this guy. Now this guy is active. So he will go and phosphorylate this guy. Now this guy is active. And that's how we're kind of passing the information along. Sort of like handing off a baton if we're doing a relay race. The baton that you're handing off is oftentimes a phosphate. Okay, so this is reception. If you were talking about reception on an AP Bio test, you have to let them know that my receptor that's receiving the ligand has the right physical and chemical fit. So it has the right physical fit, sort of like a puzzle piece or a lock and a key. There might be some flex in there. but it also has to have the right chemical fit. If this receptor has a bunch of negatives and the ligand has a bunch of negatives, that won't be the right chemical fit. They would like repel each other. So you have to be the right chemical and physical fit. Excuse me. Okay, so once that happens, then we have transduction. That's the handing off of the message, the baton. But we also have amplification. So the way that amplification works is to add in on this diagram that particular molecule right there, that protein, might activate many of these triangles. So let's say it activates 20 triangles. So I have one original message that activates one protein, and then that activates, let's say, 20. And then let's say each of those activates 20, and each of those activates 20, and each of those activates 20, and 20, and 20. Well, now by the time we get to the end, we have 20 times 20 times 20 messages. So just after 1, 2, 3 rounds of transduction, we have 2, 4, 8,000 messages out there. We just amplified the signal from one original chemical to 20, 400, 8,000 messages, and so forth. So we can also amplify the signal so that, because these guys are just diffusing along, now they get to where they're going quicker. And then lastly, you have to respond. So make sure if you're writing an essay that you give an example of what a response might be. Don't just say, and then the cell responds. Well, yeah, but how? On an AP Bio test, they'll probably usually give you an actual scenario. So if this was insulin or something like that, insulin is a protein. And so proteins, we'll talk about why in just a second, but proteins tend to work this way with membrane-bound receptors, not an internal receptor. And so whenever insulin does this, which does this, which does this, which does this, you might say like, oh, and so in the end, insulin will lower my blood sugar, or insulin will pull sugar out of my bloodstream and shove it into cells to feed them. So don't just say the cell responds if you have enough information to tell me like what that response is. If this was growth hormone, you'll say, well, then the cell is gonna start, doing cell division because it's getting a signal that says grow. Here's what's kind of interesting. Well, actually, before we get into that, we do notice a few kind of common things between these. So generally, if you look at all 20 amino acids, we might have seen a chart of that on our very first video. About half of the amino acids are polar. I want to say I think it really is right about half. It's like 9 of them, 9 or 11, something like that. It's almost like the ones that are essential and non-essential. That's also 11 and 9. But about half are polar. So in general, if this is a protein or an amino acid based signal, odds are, you could just put together a string of non-polar amino acids. So that's possible. But in general, by chance you'll have several polar ones. Usually on a protein, the non-polar parts are in the middle and the polar parts are on the outside. And that's why that protein is stable in water, because it's surrounded by polar water. And so now those polar edges are fine with it. Kind of like back to cells. The reason that they form the shape that they do is because the polar parts are fine touching water. The non-polar parts are kind of sequestered in the middle so that they are avoiding water. They're hydrophobic. Okay, so generally these are proteins. So let's say that I gave you the example insulin. Pretend that you had no idea if insulin was pretend that you never heard of it. Or if I said myostatin. Well, it doesn't necessarily work as a hormone like this, but myostatin is a real thing. If I told you it was a hormone, it's not, to my knowledge. If I told you that it was a hormone, then myostatin, you'd be like, well, it ends in IN. If something ends in ACE, it's a protein that's an enzyme. If it ends in IN or EN, it's a protein. So that would mean that it would work on this pathway. So if I said blah, 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 N, you'd be like, all right, that's a protein. It probably works on this pathway. Not all, but many of the hormones that are able to slide through the cell membrane are steroid hormones. That's because they are cholesterol based. So if I go back and look at the chemical formula, let me pull up a picture really quick. If I look at the chemical formula of cholesterol or estrogen or testosterone. So cholesterol has this, I know this says testosterone, but cholesterol has this hex pentagon shape to it. So whenever I'm looking at this right here, so here's estradiol, estrogen, hex pentagon. Real cholesterol has like a whole bunch of CH3s or CH2s and then at the end CH3s coming off the top. But if I take a cholesterol and I tweak it, then I can make estrogen or testosterone. Let me show you another example picture. Let's go to body systems, endocrine. If you watch the endocrine videos, you've seen this picture. So for example, there's cholesterol. Again, hex pentagon and a stair step. And you'll notice there's no oxygen in the corners of those, so we don't think of them as carbohydrates. And then these are all steroid-based hormones. So you can see that they all have that same shape, that hex pentagon. And then the side groups hanging off of them are different. Another thing you'll notice though is the name. Aldosterone, progesterone, estradiol, testosterone. So take the word cholesterol, and if it kind of has that ostero group in it, an ester group, we get into those more in like an organic chemistry class. Then you're like, all right, this hormone, if I said blah, 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 estro, blah, 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 you're like, estro. That's probably a steroid hormone, which means it came from cholesterol, and cholesterol is nonpolar. So that means, for example, this guy right here, testosterone, can just slide through the membrane, hit the internal receptor, and go and do its job. Okay, from here, a little bit of foreshadowing for our next set of topics. We'll be getting into cell cycle and cell cycle regulation. Here's a little bit of some cell cycle regulation. I have all these proteins that can manipulate each other. And so that's what I have up here. An arrow means that you are activating the next protein and turning it on, so to speak, which activates the next because there's an arrow, which activates the next because there's an arrow. So an arrow is saying go, go, go. What would that mean? That would mean stop. So let's say that this guy is telling my cell to grow, and this guy is telling my cell, don't grow. It wouldn't make sense to have both of these firing at the same time. The example I've given in class is if I had a crew that was specialized in digging holes, and then I had a crew specialized in filling holes, and I was a cable company and I was gonna like dig a hole, put in some new fiber optic cable, and then fill it up, it wouldn't make sense to send out both sets of workers at the same time, because as this group is digging the hole, this one's filling it up and I'm not getting anywhere. So when it's hole digging time, I turn off all the proteins that are fillers, and the only proteins that are on are the hole diggers, so they dig a hole. Then I go in, I drop in the cable. Then I turn off all the guys that dig the hole, I turn those proteins off, and I turn on the ones that say fill the hole, and now I would fill the hole. And so that's what's happening right here. As I go along here, if this pathway is on, he might have a side pathway that inhibits this guy. If this guy says grow, and this guy eventually says no, then if the answer is no, you also turn off the one that says grow or go or whatever. And so that's what we're going to be seeing in the cell cycle unit, is all of these different things manipulating each other. For example, we might get a signal from outside of the cell that says like, hey, analogy of course, but like, hey, you're 10, it's time for a growth spurt this summer. But then inside the cell, there are other proteins that are monitoring, like, do you have enough calories that you're getting every day? Do you have enough amino acids to build proteins? Do you have enough calcium to build bone? And if the answer is, I don't have calcium, I don't have enough calories, I'm only getting 100 calories a day, and I don't have enough amino acids, then they should stop that signal. I could have an external signal saying, hey, it's time to grow, but I might have some internal proteins that activate based off of some nutrient. And so as long as everything's fine, maybe they're like this. But if all of a sudden we find out that I'm low on calcium or I'm low on calories, then this pathway will activate and say, stop it. Stop it, if this was a go pathway. So hopefully I didn't get things too confusing there at the very end. Our next topic will be cell cycle. Again, after this video, you ought to be able to do a good job at 2010 number 1 and 2018 number 2. 2018 number 2 does have a little bit of immune system antibody stuff in there, so that might throw you just a little bit of a curveball, but don't sweat it. Do your best.